Hello, my name is Austin. This is my genetics exam video, and firstly we're going to look at the structure of DNA. So up here on the top left we have a phosphate group, and one thing that's important to realize is this oxygen here is negative, so this makes the entire molecule negative. Next we have the ribose sugar, and so the important carbons we need to know are the 1, 2, 3, and 5. The first carbon binds to the nitrite, or the base. The second carbon determines whether you're DNA or RNA based on if you have an OH or just an H. The third carbon will end up binding to the next phosphate group in the chain, and then the fifth carbon will bind to the first phosphate group. So this is kind of just a molecule of DNA. So next we're going to kind of look at the different bases you can have. You either have purines, which are G and A, and the pyridines, which are C and T, and U, but we're not doing RNA, so we don't need to look at U. So as we know, A and T bind together, then the reason that these are highlighted is because there's some hydrogen bonding between those two. And the G and C, the three here, also hydrogen bonding. And this kind of is the reason why the two bases line up together. So as we look at a bigger chain here, we can see this hydrogen bonding and the base pairs lining up in the middle. But something that's important to realize is that these strands are anti-parallel. Up here we have the 5N because it's the fifth carbon as it goes down to the third carbon. And then on the other side, it's the opposite way. And that's kind of why it's anti-parallel. Next, we have this not really perfect structure of DNA, but it is better because of something called base stacking. So all the bases will line up in the middle, and this is important because the bases and the sugars are not hydrophilic. They do not like to interact with water, but the outside, these phosphate groups are because they are slightly negative. And this stacking forces the water to interact with the hydrophilic phosphate groups, which are shown here. Nextly, we just have this a little bit better. This is probably the best drawing I have of DNA. We have these grooves, minor and major grooves. There's just kind of the space in between. A protein would much better be able to fit in here and interact with the DNA molecule than here, so that's why it's major. Also something to identify is the handedness of the DNA. And since this DNA molecule kind of goes up and to the right, as you can see here, then it would be right-handed. And the last part of the video was talking about how salt will increase or decrease the melting point of DNA. So as we know, all these phosphate groups are negative, and if we add a salt, there would be these positive ions that are in there, and they will be able to cancel out these negative charges, which these forces will also cancel out. And the reason that's important because the two same negative charges will repel each other, and they will kind of decrease the stability. But if we are able to shield or get rid of or eliminate these negative charges, then it increases the stability of the DNA molecule, which will then increase the overall melting temperature of the DNA molecule.